Hello and welcome to a new episode of the Medical Microbiology series. In this episode, we will continue to discuss Pseudomonas aeruginosa and cover the practical work, how to confirm the identification, and how to differentiate it from other gram-negative bacilli. P. aeruginosa is a gram-negative bacillus which appears pink following gram staining. It is also motile due to the polar flagella present on either side of the bacterial cell. This motility can be visibly demonstrated using the motility test. This is done by preparing a semi-solid agar media which is then stabbed with an inoculated needle. If the growth is only observed around the stabbed area, this is a negative result. If the growth has diffused and turbidity is observed in the test media, then this indicates a positive result. If you're interested in this, a video of the test can be viewed on our channel with further details. Another characteristic of Pseudomonas aeruginosa is that it is an obligate aerobe which is clearly noticed when the organism is injected into a liquid broth medium. It will only grow near the liquid surface which is exposed to the air because it's unable to grow in anaerobic conditions. P. aeruginosa is also catalase and oxidase positive. The oxidase test in particular is critical to identifying Pseudomonas aeruginosa and differentiating it from the Enterobacteriaceae family, the largest family of gram-negative bacilli, which contains E. coli, Salmonella, Shigella, Klebsiella, Proteus, and more, over 100 species in total. Because of this, we do the oxidase test before using API biochemical tests to determine whether it's Enterobacteriaceae or not. This allows us to determine whether to use the API 20 Enterobacteriaceae kit or the API 20 non-Enterobacteriaceae kit. P. aeruginosa does not typically ferment carbohydrates or sugars, and so it appears exactly like the control samples on API strips without any changes in the media for triple sugar iron agar, which contains glucose, sucrose, and lactose. However, it can ferment glucose, the simplest carbohydrate, if it is available through the aerobic pathway. Aerobic glucose fermentation or oxidative fermentation can be tested by inoculating a media that contains glucose, known as oxidative fermentative media, with the test organism and exposing the tube to aerobic conditions. A comparative tube should also be prepared under anaerobic conditions using oil to coat the surface. If turbidity and growth is observed in the aerobic but not in the anaerobic conditions, then this indicates that the organism can oxidatively ferment glucose like Pseudomonas aeruginosa. P. aeruginosa can grow in different simple media due to its ability to survive despite a lack of nutrients. So it can utilize tryptone soy agar, in which it appears as irregular, large, opaque coloured colonies that turn to a pale green around the third day of incubation, because this media lacks certain minerals like magnesium and potassium, which stimulate the appearance of pigments more quickly. On MacConkey agar, it produces colourless colonies because it cannot ferment the lactose present in the media, which is why it resembles the non-lactose fermenting group of Enterobacteriaceae like Salmonella, Shigella, and Proteus, although these are easily ruled out by the previously mentioned oxidase test. There are several media that can stimulate the production of pigments in Pseudomonas aeruginosa, such as cetramide agar, which contains both magnesium and potassium that stimulate the production of pyocyanin and pyoverdin pigments, giving the bacteria a fluorescent green appearance. It can even glow in the dark, especially when exposed to UV light. Pseudomonas aeruginosa can also grow on Cled and Muller-Hinton agars, where it appears as blue-green colonies. On blood agar, it displays beta hemolysis, having the ability to lyse red blood cells completely. This is easily noticed when the Petri dish is held in front of a light, where the clear zone surrounding colonies is more obvious. Other biochemical tests used to confirm the identification of Pseudomonas aeruginosa are citrate utilization, nitrate reduction, and arginine hydrolysis, which should all show a positive result. Now we're going to talk about virulence factors and how this organism shows high resistance to antibiotics. P. aeruginosa is a member of the escape pathogen group, which is the most widespread group according to its multi-drug resistance. P. aeruginosa in particular is intrinsically resistant to some beta-lactams, aminoglycosides, and quinolones due to low outer membrane permeability. The proteins present in the cell wall don't allow the passage of antibiotics to the inside of the cell. Porin proteins and efflux pumps are two examples of such proteins that regulate the inner environment of the microbe through the removal of toxic substances like antibiotics, products of metabolism, and quorum-sensing molecules. P. aeruginosa may also be adaptively resistant to some antibiotics through its ability to form biofilms, which is characteristic of this species. Biofilm formation acts as a barrier between the bacteria and the outer environment and contributes massively to multi-drug resistance in this organism. Quorum-sensing molecules are sensory signals through which the microbes communicate, which helps P. aeruginosa to resist antibiotics. Through this mechanism, bacteria are able to sense changes in their external environment and communicate these changes through those sensory molecules to other cells in the biofilm, thereby allowing it to adapt and survive. Pseudomonas aeruginosa is also able to secrete molecules known as siderophores, which collect iron from the surrounding environment. They also produce a pseudocapsule which protects it from desiccation and phagocytosis, as well as facilitating adherence to other cells with the help of pili present on the surface of the bacterial cell. These pili also contribute to the transfer of genetic information through movement of plasmids which may encode antibiotic resistance. P. aeruginosa can produce both endo and exotoxins. Endotoxins are formed from the lipopolysaccharide present on the cell surface, which is secreted after cell death. Exotoxin A stops protein synthesis and is responsible for tissue necrosis. Thank you for watching. I hope this video has helped you to gain some useful information about Pseudomonas aeruginosa.